Hello everyone, a very good evening to all of you. So today we are going to start with a question discussion from Unit 4 and that is cell signaling. So can you please confirm? Am I audible to all of you? Screen is also visible, right? Okay, let's start with the session today. Okay, good evening, Sunshine. Okay, fine. So, to begin with, I hope so you have studied cell signaling, right? And we will discuss some of the pathways and how it is related and how the questions are coming in CSIR exam. Good evening, Reshma and Rahul, Surendra. Good evening. Okay, fine. So, moving on to our first question, a bit a simple question. Let me see how many of you can attend this question. Which one of the following combinations must be present in a steroid receptor that is located in the cytoplasm? Nuclear export sequence with leucine zipper, NES with zinc finger motif, NLS with zinc finger motif, NLS with leucine zipper. What is the correct answer over here? Quite an easy question, I suppose. They are just asking you the location of or which combination will be present in a steroid receptor that is located in the cytoplasm. Okay, I can see answers coming up. Reshma, Surendra, Priya, Rahul, Sunshine. Okay. Any other answers? Anyone else would like to try? Sampurna. Good. Any other answers? Anyone else would like to give a try? So, the correct answer, as you all mentioned, okay, Aishwarya, is your option 3, NLS with zinc finger motif. Now, can anyone tell me which domain you will find zinc finger motif? Are you aware of the structure of steroid receptors? The model for steroid receptor, anyone aware? In that case, let me know where is this NLS located and where is this zinc finger motif located. Okay, within the PPD domain. And what about NLS? What about NLS? Nuclear localization signal. We can easily omit these two parts if you are confident with zinc finger motif, right? And since it is located into the cytoplasm, it needs to enter inside the nucleus. So definitely it will be having NLS over there. When this NES will be working, when any protein from the nucleus has to be exported out. So since they are saying that it has to be incorporated into the nucleus, so you will be having NLS over here. Yes, where is NLS located? DNA binding domain, so you are having zinc finger motif, that is correct. What about the NLS? Where it should be located? I'm asking within the receptor where this NLS is located. Okay, let me show you the model of a typical steroid hormone receptor. So what does it say? So since every receptor is a protein, So obviously it is having an N-terminal domain and a C-terminal domain. In between, you are having 4 regions. The first region is known as variable domain, where you will find interaction with transcription factors. And why do we need transcription factors? In order to generate mRNA. Because after that only you'll get the proteins, right? Then the second domain is DBD. So this is your DNA and this is your zinc finger motif. So they will be bind to the DBD domain. So do we have any specific sequence within the DNA where the zinc finger motifs bind? Do we have any specific sequence within the DNA where this DNA, where the zinc finger motif will bind? I mean the steroid receptors will bind. Yes, you are correct. That is ligand binding domain only you will find the NLS. That is true. Okay. Now my next question is, what is the specific sequence elements present within the DNA into which your zinc finger will go and bind? Activation factors is a part of your steroid receptor. Okay. I am asking within the DNA, what is the sequence? Because your receptor has to bind over here. Absolutely correct. We are having hormone response elements. Okay. Then, when a ligand goes and bind to the receptor, obviously it will undergo dimerization. So that is this domain. Now, what is the significance of dimerization? What is the significance of dimerization? Why do this receptor ligand upon binding undergoes dimer? Some reason should be there, right? Without any reason, nothing happens in this world. So similarly, if a dimerization is occurring, there should be a reason behind it. What is that reason? Yes, only upon dimerization only your NLS will be exposed. Then only it can enter inside the nucleus, binds to the HRE sequence within the DNA and carry out transcription. Okay, and this is your ligand binding domain. Now, if we look into the binding, how does it binds? Via, so you will be having a dimer. Into that, your DNA will be there where you'll be having hormone response elements. So via zinc finger motif, 2 zinc finger motifs will be there because you are having a dimer. So via zinc finger motif, it will go and bind to the hormone response elements. Okay, so this is how the construction of a typical steroid hormone receptor works. Now, next question to you. Which one of the following statements about the nuclear receptor is not true? Receptors are always cytosolic, where they remain associated with HSPs and have variable ligand binding domains in the N-terminal region. Receptors will be having a repeat of C4 zinc finger motif. Receptors could be either homodimeric or heterodimeric, and in the absence of the ligand, it represses the transcription. Then, receptors have a unique N-terminal region of variable length and may contain an NLS present between DNA and ligand. Good evening, Sharda. So, what should be the incorrect statement over here? I always say, whenever you read a question, read the question thoroughly. What is the question they are asking, whether they are asking the correct statement, incorrect statement, true or not true. Screen is not visible. What about others? Is my screen is visible to everyone? Kindly confirm. Is my screen is visible to everyone? It's visible, right? Okay. It's visible, Sayanti. Check your internet connection once. Okay, fine. Yeah, what should be the answer over here? Okay, what should be the answer over here? I can see some of you has given the answer as 1. Anyone else would like to give the answer? Is it that much tough question? I don't think so. 4, I got an answer. 1 and 4, I got as an answer. Any other inputs from anyone else? Okay. So the correct answer over here, I mean to find out the incorrect statement over here is statement 1. Now, whoever has told me statement 1 is incorrect, can you tell me why? Why the statement 1 is incorrect? I guess everyone will agree it is having a zinc finger motif, right? And why it is homodimeric and heterodimeric? Can anyone tell me why statement 1 is incorrect? Yes. Statement 1 indicates what? The receptors are always cytosolic, where they remain associated with HSPs and they have a variable ligand binding domains towards the N-terminal region. Yes, absolutely correct. This particular line is incorrect. Why? Because we are having two types of receptors. Do you remember? Type 1, type 2 intracellular receptors. Anyone able to recollect? Two types of receptors, type 1 and type 2. Remember? Okay. So here it is. See, when we speak of intracellular receptors, we are having two categories. Type 1 is present in cytoplasm, usually combined with heat shock proteins. All the sex steroids, glucocorticoids falls under this category. And they are responsible for forming homodimers. That means same molecule will undergo dimerization. When we speak of type 2, these kind of receptors are present in the nucleus and they are directly bound to DNA. Example includes our thyroid hormones, vitamin D. That means vitamin A is also coming over here. And in this case, we will find that receptor to ligand interaction can be a homodimer as well as a heterodimer. Okay, so these are the examples. Corresponding receptor will be having the corresponding ligands. So androgen receptor, estrogen, progesterone, corticoids, all of them belongs to type 1 receptor. That means their location will be in the cytoplasm. And they will be bound with HSP forming homodimers. Type 2 receptors, if you see, you will be having vitamin derivatives, vitamin A, D, retinoid, thyroid hormone receptors. So the location will be in the nucleus. Bound to directly DNA. It can be homo or heterodimers. Okay, got it? Very good. So let's check out the signaling. Do you know how the signaling takes place in type 1 and type 2? Do you know how the signaling takes place in type 1 and type 2 receptors? Very good. See, this is your type 1 and this is your type 2. So what is the basic difference in terms of signaling? If you look into the image, see, this is your receptor having LBD and BBD. Okay. So, when you don't have a hormone, the cloud-like structure which you see, that is your heat shock protein, is bound with the receptor. When this hormone comes and binds to the receptor, the HSP will be released. Undergoes dimerization. Via nuclear pore enters inside the nucleus and binds to the hormone response elements present within the DNA. So the next step is to carry out transcription. So for that purpose, RNA polymerases, co-activators will be recruited that will help in the transcription. And since we know that protein synthesis takes place outside the nucleus, it goes out and undergoes protein synthesis. So that is your type 1. Now, when we speak of type 2, what happens? As I told you, the repressor is already located within the DNA only. So instead of HSP, you are having the co-repressor present. So when you don't have a hormone, co-repressor represses the transcription. When this hormone comes and binds to the receptor, you can find repressors are moving out. Polymerases and co-activators will combine and added over here in order to carry out transcription. That will move out of the nucleus via nuclear pore to carry out protein synthesis. Is there any confusion in these two pathways? Type 1, type 2, are you all able to understand? The basic difference which you need to remember here is these are cytoplasmic and this is nuclear. Then HSPs are bound. Here co-repressors are bound. What is the third difference? They will form homodimer. And in this case, you can see it is heterodimer. Clear to all of you, I guess? Everyone is able to understand? Very good. So this is how the binding takes place. So this is your DNA, okay? And you are having the zinc finger motifs. Since it's a dimer, so 2 on each, all total you are having the 4 that will go and bind to the hormone response elements. The connecting link between the ligand binding domain to that of a DNA binding domain is hinge region. Okay, so like that it will carry out transcription. Yes, it may undergo homodimer. In this case, it may undergo homodimer and heterodimer. As you can see, your thyroid receptor is combined with the retinoid receptor. So in that case, what happens? It can form a heterodimer. If thyroid to thyroid receptor is present, that is also possible. It will undergo homodimerization. Okay. So this is the part of nuclear receptor. And once upon a time, this nuclear receptors used to be very important from examination point of view. They used to ask questions from here in different forms. So please focus on that. Next question. Acetylcholine is a potent neurotransmitter that is released from the neurons. After their release into the synaptic cleft, it combines with nicotinic receptor molecules. So the interaction of acetylcholine with nicotinic receptor produces large transient increase in the permeability of membrane that will allow ions to pass through, thereby providing the signal transduction of nerve impulse. So acetylcholine receptor by nature is what? Ligand-gated ion channel, ligand-gated anion channel, voltage-gated cation channel, or voltage-gated anion channel. This is a 4 marks question. Very direct question. The only thing is that you have to be sure of what kind of channels are there open. Yes? Okay, very good. As I can see, majority of you has given as answer 1. Anyone else? The thing is that you just have to remember what is a cation anion, and since it's already the ligand is given, so we can always say it's a ligand gated channel. We don't have any role of voltage over here. Okay, fine. Anupam, Kaushalya, and Ashu. So the correct answer over here is ligand gated cation channel. Now, on to this question, I want to know the forms of receptor. Neurotransmitter goes and binds to the acetylcholine receptor, they said. It exists in how many forms and what are they? It exists in how many forms and what are they? Tell me the structure of an acetylcholine receptor. Can anyone able to say? Structure of acetylcholine receptor. 3 forms, absolutely correct. What are the forms? What are the 3 forms of this receptor? Yes, absolutely correct, Shakti. So here is the image for you. You are having a resting phase, an excited phase, and a desensitized phase. What do you understand by desensitization? What do you understand by desensitization? Open, closed form, we can say we don't have acetylcholine present over here. So the gates are closed. Once your acetylcholine goes and binds to the receptor, it opens the ion channel that will allow the passage of ion. So you call it as a excited phase, or the gates are open. Continuous stimulation leads to what? Desensitized phase. Now, what is this desensitized phase? You can find what is the difference between this and the resting phase is that here the gates are closed, keeping the ligand inside. Okay, and here you don't have a ligand, so that is your resting phase. Desensitized, your you can also call it as a kind of termination process. Okay, one way of terminating a signal transduction pathway. So after some time, slowly, gradually, this acetylcholine will be released and the receptor will come back to the first phase, ready to take up another set of reactions. Yes, and I guess you all might be aware the receptor is made up of 5 subunits. That is 2 alpha, 2 beta, and 1 gamma. So it is in these 2 alpha subunits your acetylcholine goes and binds. Okay. And what kind of binding you can see? We call it as a cooperative binding. Why do you call it as a cooperative binding? Because binding of one acetylcholine at one site influences the activity of another acetylcholine to go and bind over here. Okay, so these are the three different forms of acetylcholine receptor. Now, let's check it out how exactly the signaling takes place here. So, if you see, this is the axon of presynaptic neuron. So let me enlarge this image so that it will be more clear. Okay, and this is your cell body of postsynaptic neuron. Okay, now the junction between the pre and postsynaptic neuron is known as synaptic cleft. So what happens upon stimulation? Voltage gated sodium channels will be opened. Okay, so more number of sodium channels are open, which induces voltage gated calcium channels to open. Now, what is the purpose of voltage gated calcium channels? Because we have our ligand, that is acetylcholine, present within the secretory vesicles. So this calcium channels helps in the fusion of the secretory vesicles to the synaptic cleft, which will release the corresponding ligand, goes and binds to the receptor that is present in the postsynaptic neuron. So again, the same kind of action potential takes place, and like this, the signal will be transmitted to the next neuron. And it keeps on transmitting unless and until it reaches a saturation zone. Clear? How we are going to terminate this pathway? Of course, one way is desensitization. Do we have any other way to terminate this ligand-gated ion channels? Do we have any other way of terminating this acetylcholine mediated signal transduction? Absolutely correct, Reshma. We are having acetylcholine esterase. Now, what this will do? This will help in the degradation of ligand. So, when the ligand itself is degraded, so there won't be any interaction between ligand to the receptor. So, at the same time, signal will be terminated. Okay, so that is your second question. Third question. Next, moving on to the fourth question. An action potential was generated on a nerve fiber by a threshold electrical stimulus. When you are providing a second stimulus, no matter how strong it was, during the absolute refractory period of the action potential, the nerve fiber was unable to generate a second potential. And these were the observations seen. A large fraction of potassium channels was voltage inactivated. Critical number of sodium channels required to produce an action potential could not be recruited. Large fraction of sodium channels was voltage inactivated, and the critical number of potassium channels required to produce an action potential could not be recruited. You have to identify the correct combination over here. Now, for this, you must be familiar with the graph of action potential, resting membrane potential. Yes, what should be the correct answer over here? Reshma and Sunshine has given the answer. How about others? Read the question thoroughly. Remember, where is the refractory period present? And what happens? Okay, I got a different answer, 4. So, 3 and 4. How about others? Sampurna, Aishwarya, Sharada. Okay, okay. There is a kind of what to say confusion between option 3 and option 4. I can see. Okay, option 2 is also some of you are saying. Ashu. Okay. Remember what is a threshold electrical stimulus? What is an absolute refractory period? What are the stages of action potential? Then give the answer. That's it. Anyone else would like to give a try to this question? What do you mean by absolute refractory period? And where does it come? Can anyone tell that? What do you mean by absolute refractory period of an action potential, and in which stage you will find this period coming, this refractory period coming? Okay, there is some confusion for sunshine. Between depolarization and repolarization event. Okay, see what is the graph over here. So the correct answer is C and D. I can see many of you has given the answer as 4, and you are absolutely correct. A large fraction of sodium channel was voltage inactivated. Critical number of potassium channels required to produce an action potential could not be recruited because it is just in the starting event. Right? See the graph. What do you mean by resting membrane potential? And how can you differentiate it between action potential? Can anyone tell me? Resting membrane potential to difference between action potential. I think this is the part of unit 7 as well under nervous system. So you might be more familiar with this topic. So I just went for superficially with respect to signaling pathway. Yes, Ashu. Resting membrane potential basically lies in minus 70 MV. Who is responsible for maintaining this resting membrane potential? Who is responsible, or how minus 70 MV is present for resting membrane potential or maintained at resting membrane potential? Maybe we have something. Otherwise, how the membrane becomes negatively charged from inside? Yes, we are having sodium potassium ATPases. Since more number of positively charged ions goes outside, so the inside becomes negatively charged. Yes. And what about action potential? It's a combined action of depolarization to repolarization. So when you get a stimulation from the resting potential, it goes to threshold potential. And what is the significance of threshold potential? From here, it will only go up, it will not go down. So as you can see, more number of sodium channels opens up. That means sodium ions comes inside the cell, making up to plus 30 MV. Beyond which, it will not open. Slowly, gradually, potassium channels will open. And potassium ions, what they are doing? They are moving out of the cell. So that stage we call it as a repolarization. Now, when more number of potassium ions are going outside, what happens? The membrane potential goes beyond to minus 70 MV, which we call it as an hyperpolarization event. right? Going beyond minus 17. After that, what happens? Your potassium channels close. So it comes back to RMP. The difference in blue color you can identify. In between, you are having two refractory periods. Absolute refractory period that comes between D and repolarization. Okay. So what happens if a stimulus is provided and one action potential is moving, unless and until it reaches and comes back to the RMP, it will not start another action potential. So no matter how much strong a stimulus you give at this junction, it is not going to revert back. It will go continuously. The membrane will not respond to any kind of stimulation. What is relative refractory period? Once it reaches the RMP, after hyperpolarization, once it reaches the RMP, you need to provide a stimulus that is larger than the normal one. So this leads to what? Another action potential. Okay. Is this graph clear to all of you? The events, everything, the terminologies, when is channeling or channel opening, channel coming inside, I mean ions coming inside, ions going outside, is these things are clear? Or is there any confusion in this graph? Clear, right? Okay. One more term is also there, you might have heard, hypopolarization. Do you know what it is? Hyper means, as I told you, going beyond minus 70 MV. What is hypopolarization? Hypopolarization means what? Hypopolarization means what? That is, okay, negative potential you are saying. Hyperpolarization is actually related to negative potential, Reshma. In that case, what should be hypopolarization? Just think the other way around, you'll get the answer. Hyper related to negative potential. That means what is hypo? Absolutely correct, sunshine. In fact, I can say that this depolarization is otherwise known as hypopolarization. Okay. So, yeah, it will be less. So, remember the terminologies are important from examination point of view. And of course, if you understand the terminologies, then you can easily plot a graph like this and you can get an answer. Okay, so like this, statement based questions you can expect. Okay, so these are negative and this is positive. Okay, next. Very easy question. Which of the following is not a second messenger here? Cyclic GMP, diacyl glycerol, inositol triphosphate, phosphatidyl inositol. So at this junction, you must know different examples of second messengers. I think everyone should answer this question. Yes, you can see a lot of answers coming up. Anyone else? Yes, Sharda, Sadhya, Sunshine, Anchal, Reshma, Rahul, Sampurna, and Aishwarya. Okay. Anyone else would like to give your inputs? Okay, Sayan. I expected some more answers. What about others? That much easy question is this. Now my motto to share this question with you to make you aware regarding the second messengers. As you know that second messengers is an integral part in case of a signal transduction pathway. Can anyone tell me second messengers lead to the activation of which protein? Next, in a pathway, if you see second messenger leads to the activation of which next protein? So if I mention as a second messenger over here, okay, who is activating second messenger and second messenger is activating what? Yes, second messenger leads to the activation of protein kinesis, and your effector protein is the one which activates the second messenger. That is the pattern. Okay. So the correct answer over here is phosphatidylinositol. So you should not get confused with these two terminologies. Now, can anyone tell me cyclic GMP acts as a second messenger in which signal transduction pathway? Cyclic GMP acts as a second messenger in which signal transduction pathway? Let me see how many of you can answer this question. You got my question, right? Now you need to tell me all this. So first, let's start with cyclic GMP. SH2 is not a protein, Sayanti. SH2 is basically a domain present in the anchoring protein. Okay. Yes. One example I got from Sampurna, ANF. What about, do we have any other pathway where cyclic GMP plays a role? Yeah, the same thing, Sunshine. GC, ANF, that is correct. Do we have any other pathway where CGMP plays an important role? Think out of all the pathways. Is there only ANF present? So this comes under the category of receptor tyrosine kinase pathway. Right? That's it. Cyclic GMP is present only in RTK pathway. Very good, Sampurna again. We are having, yes, Reshma, GPCR pathway, that is your GT pathway, that is related to your rhodopsin vision. Okay, very good. Now, what about diacylglycerol? And this is your IP3, right? So this is your DAG. So what about these two? IP3 and DAG acts as a second messenger where? In which signaling pathway? Again, absolutely correct, Sampurna. So we have GQ pathway where we have PLC which breaks down to give rise to IP3 and DAG. What is the function of IP3 here and what is the function of DAG here? Yes, Sunshine. What is the function of IP3 and DAG? Gradually, I'm getting a less response. What happened? Suddenly, you are not able to recollect the thing. Suddenly, you are not able to connect the thing. Yes. What is the function of IP3 and DAG? Absolutely correct. Surender, IP3 is responsible for releasing your intracellular stores of calcium ions, where it is stored in the endoplasmic reticulum. DAG combines with increase in the cytosolic calcium level to activate your protein kinase. Which protein kinase? Which protein kinase will be activated here? You are having protein kinase A, B, C, and G. Which protein kinase is activated here? That is correct. Calcium release to sahi. That's correct. We are having protein kinase C. C for C. You remember? C for C. That is a code word. Okay. So that is about your second messengers. Okay, this is just a reminder that we are having ion exchange chromatography hands-on training on 16th September 2023 at 10 a.m. IIC incubated lab, that is protein to science. So both online and offline facility is available. Those who are staying in Bangalore can directly go into the lab and you can actually see live. Otherwise, if you prefer online, you will get a Zoom link and you can attend it over there. Reminding you for another one, that is we have 2 days workshop, that is hands-on workshop for fermentation. So any one of you who is interested with bioprocess, fermentation and all, you can attend by bios. And this will be taken place on 27th and 28th of September. 2 days will be there. So here also you have both online and offline facility. So depending upon your choice, you can choose one of them. So if you have any further queries regarding this workshop, you can call to this toll-free number, either of this toll-free number. So we are having limited seats. So just make sure that go through the portal, go through the things what we are going to discuss in this and attend if possible. Next. Now coming to receptor tyrosine kinase. It forms a large and important class of cell surface receptors whose ligands are soluble or membrane-bound protein or peptide bonds. Which of the following statements is considered to be incorrect over here? All RTKs are transmembrane proteins with extracellular ligand binding site and a cytosolic too. Binding of ligand causes most RTK to dimerize. Cytosolic tyrosine kinase phosphorylates a distinct set of tyrosine residues in the cytosolic domain. And adapted proteins and enzymes involved in signaling pathway binds to different tyrosine residues via SH2 domain. Can anyone able to give this question's answer? Now here also you have to identify incorrect statement here. What should be the incorrect statement out of this? Yes, Sampurna and Anchal has given the answer. Sayanti. Sunshine. Very less answers this time. Previous question, so many answers I got. Reshma, Surinder. All are opting for option 3. Okay. Anyone else would like to give Any other answer apart from 3? Anupam also opted for option 3. Only one answer I got. Musta monomeric. Okay. But here they are asking the incorrect statement only, right? Anyone else trying to give the answer? These are the features of your receptor tyrosine kinase pathway. I hope so. You all are aware of different domains. What are apart from SH2? Do we have any other domains present in adapted proteins? Can anyone tell me, apart from SH2 domain, do we have any other domains? Okay, SH3 is there. Apart from that, apart from SH2 and SH3, apart from SH2 and SH3, okay, let me give you the answer. As mentioned by everyone, I think I don't have to give any explanations over there. The correct answer over here is 3. That means that is the incorrect answer among the all. So, as I told you, all RTKs are considered to be transmembrane proteins. That means they are tethered to the membrane. So they will be having the extracellular ligand binding site and intracellular cytosolic domain. So it is in the cytosolic domain you will find the kinase activity present. That means it could be your tyrosine kinase, serine threonine kinase, anything. Then it's okay. I will remind you. Binding of ligand causes most RTK to dimerase. Actually, you provided the correct explanation, Sunshine, but you could not give the right answer. But no problem. See why your answer is incorrect. As you told, most of this receptor tyrosine kinase is monomeric by nature. Only in some exceptional cases they are present in dimer form. For example, in the insulin receptor. But majority of the cases they are monomeric by nature. So when your ligand goes and binds to the receptor, then only your tyrosine kinase will undergo dimerization. The receptor will undergo dimerization. Okay. So that's the reason statement 2 is also correct. And statement 4, it's correct only because SH2 domain is having affinity towards what? Phosphotyrosine residue. And SH3 domain, some of you has told me. It is having affinity to what? SH3 domain is having affinity to what? The way for SH2 you are having phosphorylated tyrosine residues. You told me. And regarding SH3, what it is having the affinity? Or which amino acid it will be having the affinity? Yes, it is your proline residues. Right? Similarly, like SH2, you are having PTB domain. That is also having affinity towards the tyrosine residues. And you are having pH domain, someone has mentioned. This is having affinity towards the lipid components. Right. So, why the statement 3 is incorrect? Why the statement 3 is incorrect? As I can see, majority of you has given the correct answer. Why statement 3 is incorrect? Can anyone provide me the explanation? What it is given? Cytosolic tyrosine kinase phosphorylates a distinct set of tyrosine residue present in the cytosolic domain of the dimer. Why this is incorrect? Why this is incorrect? Yes, everyone has given the correct answer. What about the explanation part? Because there is nothing as such a distinct set of tyrosine residues. If you go for receptor without kinase activity means you can say a distinct set of tyrosine residue. The reason is that there you are having the tyrosine kinase present in the cytoplasm that will be recruited to the receptor. Okay. So that's the reason you cannot say cytosolic tyrosine kinase phosphorylates a distinct or unique set of tyrosine residues. It's a normal one only. Yes. Yes. Yeah, it will undergo phosphorylation, self phosphorylation, it will undergo. Okay. And this is an image for a comparison. As we have majority of growth factors, if taken into consideration, they undergo RTK pathway. Okay, and all of them are same in terms of cytosolic region. Okay, because all of them are having tyrosine kinase activity, but what or how they vary based upon the extracellular region. If you see for insulin receptor INSR, you are having a dimeric receptor. It's already present in the dimer, but if you see others, it is existing in the form of monomers. But when a ligand comes and binds over here, it will undergo dimerization. That is one difference. Second difference is that if you see the component for insulin receptor, you are having leucine and cysteine rich domain, a combination of them. For VEGFR and PDGFR, you are having immunoglobulin domains, right? But what is the difference? Difference in the length of the chain. Okay, and here also the same way you are having like your insulin receptor, combination of leucine to cysteine. In this case, you are having a leucine along with the immunoglobulin domain, that is your neuronal growth factor. And if you take the case of a fibroblastic growth factor, you will be having a combination of aspartate or glutamate rich region along with the immunoglobulin domain. Okay, so that is the difference among different receptors in RTK pathway. Of course, I cannot say 100% of growth factors undergoes RTK. If I say this, this is incorrect statement because we have some exceptional cases. So we can say that almost 90% of growth factors will undergo RTK pathway. Okay. So I hope so you got better insight with the question discussion on where you need to focus on for the upcoming times. Am I correct? You understood all the questions, how the questions come and how you need to approach a question, how to understand a question. Is all the things are clear from your end? Yes? Okay, so in the next session, make sure that you study the majority of GPCR pathways and come to the session, specifically related to vision. Vision related questions are very much favorite in CSIR NET exam. And many a times you will come across a question based upon this GT pathway. Even last recent exam also, if you just go through it, you'll get a question out of this. But the thing is that you have to just understand the protocol, then you can just implement it. Okay. Okay. So Sure, sure. Thank you so much, everyone. So keep practicing MCQs as I tell to everyone. Keep revising the topics because you have 2 more months to go and practice MCQs regularly. Dedicate at least one hour every day to revision as well as MCQs. Stay connected with the topics which you have already done because if you keep it aside, the problem is that you will forget if you take it out after a week. That's the reason. Stay in touch with the topics by spending at least 1 hour in a day for revision and 1 hour in a day for MCQs. Go ahead with that. See you again in the next session. Thank you so much, everyone.